Hello students. In this video we'll talk about exercise 7. This exercise is an online exercise for you to do, and it relates or it compares photosynthesis and cellular respiration. These are topics from chapters 9 and 10 in our lecture textbook. So let's take a look. I'm going to look at our Blackboard site and find our exercise. So we're down here in exercise 7. Click on that and you should have or should be doing pretty quickly the pre-lab quiz, right? The documents here. Here's the pre-lab quiz, the worksheet assignment, and several videos that are also linked in the document that you'll be watching, all right? A video on photosynthesis, on respiration, and an interesting HHMI video on extreme animals and their RQ, which we'll learn more about. Okay, let's take a look at the document, Photosynthesis and Respiration Online document. All right, exercise 7. So first we'll kind of take a look at what our objectives are. We're basically going to have you use a simulation, use 2 simulations to collect data and think about what the data mean. So after completing watching videos and reading this document, then you'll want to take the pre-lab quiz by Sunday night, as usual. And then we'll read through this introduction, and we'll skim through this a minute to see what we need to do. So first of all, you'll want to watch these basic videos on photosynthesis and respiration, cellular respiration by the Amoeba sisters. You should be in lecture, you should be focusing on all the gory details of cellular respiration and photosynthesis. And there are a lot of details. I hope you're enjoying learning about them. I'm kind of fascinated by the details, but we've posted here the big picture. The big picture is the summary equation, if you will, for each of these processes. And remember, in respiration, that's the first equation here. In respiration, we use a fuel like glucose, and in the presence of oxygen, we oxidize that fuel, making a bunch of byproducts. Carbon dioxide is a waste product, water, and energy. Energy that's released is used to make ATP. ATP is what our cells use to do all their work, right? Photosynthesis, the summary equation, is essentially the reverse of this. And in photosynthesis, carbon dioxide is grabbed from the atmosphere by photosynthesizing organisms and taken in, right, in the presence of water and sunlight. So water and sunlight are contributing in this whole process to the production of sugar and a byproduct of oxygen. Now, both of these processes are complicated. So when you start talking about cellular respiration, for example, you'll focus on glycolysis, the citric acid cycle, and then the process called oxidative phosphorylation, and figure out how all of these contribute together to the production of ATP by the cell. When you talk about photosynthesis, you'll talk about the light reactions and the light-independent reactions, or as we call it, the Calvin cycle. And what happens in the light reactions? How do cells take in light energy and how do cells take in light energy to make chemical energy? How do they convert it into chemical energy like ATP and NADPH? And then the Calvin cycle, the cells grab carbon dioxide and convert it take the carbon in carbon dioxide and put it into an organic molecule. That's pretty cool too, making the sugar. Okay, there's a little bit of information about something called the respiratory quotient. When you're thinking about cellular respiration, researchers say, well, you don't always use sugar as your source of fuel. We know that because we eat all different kinds of foods, and we can use all different kinds of foods as fuel to make ATP. But we can figure out what kind of fuel an organism is using based on the respiratory quotient. The respiratory quotient is just a ratio of the amount of carbon dioxide produced divided by the amount of oxygen used. And if you are burning or using as your fuel only glucose or sugars, then this ratio should be 6 carbon dioxides divided by 6 oxygens, or a ratio of 1. That would be our respiratory quotient. If we change this fuel from sugar to a fat or to a protein, that changes the respiratory quotient. So we can get a feeling for what type of fuel an organism is using based on its respiratory quotient. And the HHMI science video talked a little bit about this. So I hope, have learned a little bit about this. Okay. In photosynthesis, we don't talk about respiratory quotients, right? In photosynthesis, we are using 6 carbon dioxides to make 3 carbon sugars, or a 6 carbon sugar. So there's 6 carbons with there's 6 carbons that are being conserved, and this always releases 6 oxygen molecules. So you get this ratio of 1 all the time. Okay. But it's interesting to think about photosynthesis and respiration and how both of these are important, because remember, plants that photosynthesize are also respiring, right? They have to take the sugars they make and convert them into ATP so that the cell can do work. All right, so for your exercise, you're going to be going online to John Darkow simulation site, and you'll be working through both a photosynthesis simulation and a respiration simulation. And so let's take a look at the photosynthesis simulation. So you can click on that simulation site and click on the tab Metabolism, Photosynthesis. And what you'll see is some background information, some references, and the simulation site itself. And that's where what we'll be looking at primarily, but the background is useful too. And remember, in photosynthesis, light energy is required, water is required, and during the process, during the light reactions, light energy is converted into NADPH and ATP. But then during the Calvin cycle, NADPH and ATP are used to help make sugar, to reduce carbon dioxide and make sugar. Okay. So in this simulation, there are all different things you can change, like is it in the light or in the dark? Is the light source a full spectrum, or is it a specific wavelength? And you should be able to change that wavelength. What's the distance of the light? Distance of the cells from the light. All right, and what are some other important things like carbon dioxide levels, NADPH, temperature? I'm not changing any of that. I'm going to run this, and we're going to take a look at what we get. Okay, what we're looking at here are a whole bunch of different products that are produced during photosynthesis. So we got an overview graph with all of the products together, and then you can click on each one of these. So during photosynthesis, of course, we make glucose. Some glucose is made. Okay, that's our end product of photosynthesis. And oxygen is released as a byproduct. So we see oxygen going up in this particular example. ATP and NADPH are made, but then used, and made and then used. So we're going to see kind of a oscillation in ATP and NADPH. Yeah. So it's made, it's used up. It's made, it's used up. It's made, it's used up. Okay. What you're going to do in your worksheet is you're going to manipulate one variable. You're going to manipulate the wavelength of light under which this experiment is taking place. So I'm going to click a minute onto our exercise here under photosynthesis. I just want to point this out. You're going to change the light wavelength. You're going to select a variable from 400 to 700 nanometers. You're going to run this at least 8 times, possibly more. You want to pick this whole range of wavelengths. You think, I wonder which wavelength photosynthesis is going to be the best at. So that's really what we're asking. And then you're going to run it at least 8 times, but I also mention here, do several runs between 450 and 480 nanometers. And that's because there are big changes in how much glucose is produced, how much photosynthesis happens in these different wavelengths. So we want to measure that, okay? And then you're going to record your data on the worksheet. So in your photosynthesis simulation, you are going to be varying the specific spectrum or the specific wavelength. So you're going to convert that to, oh well, let's reset this a minute. You're going to vary the specific wavelength. And it doesn't say what specific wavelength we're using in this first experiment, but if I run it, it tells us that the specific wavelength was 680 nanometers. So we're talking kind of reddish light, if you will, all right? And you can record the data for 680 nanometers for each of these 4 components. Then you have to change the wavelength. And like I said, vary it from, all the way from 400 nanometers to 700 nanometers, but pay specific attention, like I said, to that small set of wavelengths in the kind of bluish to green range, mostly bluish range. And, maybe you'll do like 465 or something like that. And you click go. And that's the red line this time, and I can see how much glucose is produced or how much oxygen is produced, how much ATP is made after 40 minutes or NADPH, okay? And you're going to be recording all of this information on your worksheet. So I'm going to click on my worksheet just so I kind of have a feeling for what's happening on that worksheet. So here's my worksheet. It says I can earn up to 18 points on this worksheet, an extra credit point. Woohoo, that's good. And here I am under the photosynthesis simulation, and I'm going to change the specific wavelength of light for this simulation. I'm going to vary my wavelength of light and record all of these data points. Then I'm going to add atrazine. Atrazine is an herbicide, all right? And you can learn a little bit more about atrazine on the simulation site. And I'm going to change the amount of atrazine I had. And an herbicide is going to kill plants. So I'm guessing that when I add atrazine, it's going to affect photosynthesis. That's just my guess. So if you go back to the photosynthesis simulation, you can see at the bottom that you can change the amount of atrazine that you add. And so I think I wanted you to use 680 nanometers. I'm not quite sure. You have to read that carefully. And then use either 0.5 atrazine, 0 atrazine, or 1 atrazine, and then run the experiment. Oh boy, what am I looking at? How much glucose is produced? That's not good. We didn't have much photosynthesis happening here, okay, with atrazine around. It must do some damage, so you can find out what it does too. Okay, what else do I need to do? So after I've completed this simulation, oh yeah, it talks about how I have to deal with the atrazine. Then I answer the questions on the worksheet, on my worksheet. So I've completed, so I'm looking at my photosynthesis simulation. I've completed the first tables. Oh, and I need to make a graph of the effect of wavelength on oxygen and glucose production from photosynthesis. So I'm only going to focus on oxygen and glucose. So I'm going to have 2 lines of data. Wavelength is my independent variable, so it's going to go on the y-axis. Amount of oxygen and glucose produced is going to go on the y. Did I say what? Did I say that wrong? The wavelength is going to go on the x-axis. The y-axis will have oxygen and glucose produced. Okay? Make sure you get that right. Add a good caption. Add some information about drawing your conclusions, and then we're ready to move on to the respiration simulation. In the respiration simulation, if you go back to our document, you can see what we need to do. We need to find, under our John Darkow simulations, we need to go to the cellular respiration accounting model. And it's very similar. Again, there's some information here, introduction and background information, and there's even information on the experimental setup, so we know what we're doing. That might be worth checking out before you run this. And remember, in cellular respiration, we're taking glucose and oxygen and using it to make ATP, carbon dioxide, and water, right? But we see here how much glucose is used, ATP produced, how much oxygen is used, how much NADH is produced. NADH is that electron carrier or shuttler that is produced but then used up during the process of cellular respiration. So let's take a look, and we can modify all sorts of good things here. But I'm just going to run it and see what we get. A big fat mess, right? We can look at how much ATP is produced under these conditions, or how much oxygen is consumed. So we go from 100% down to 75% or something. Okay, the metabolism of 1 mole of glucose. Okay. Now, in your experiment, you are going to modify the pH of the intermembrane space. So boy, now you have to start thinking about this. The intermembrane space is in the mitochondrion. It's the space between the two membranes. And if you've had chapter 9 material in lecture, you should understand that this pH is important. pH refers to the concentration of protons in a space, right? And a low pH means a high concentration of protons. It's very acidic. A high pH means a low concentration of protons. It's very basic. Turns out that the concentration of protons affects how much ATP can be made. If there's a gradient, a proton gradient across the inner membrane of the mitochondrion, if there is a lower concentration of protons in the matrix than in the intermembrane space, then we can make a lot of ATP when protons move through ATP synthase. So I'm going to vary the pH of the intermembrane space, and I tell you to pay particular attention between a pH of 2 and a pH of 9. If you get to the extremes, your pHs, you won't be able to, the mitochondria won't be able to do cellular respiration. So you want to focus between pH of 2 and pH of 9 on your data collection. So let's go back and take a look at this information here. So in cellular respiration, you are going to test both groups A and B are going to look at the pH of the intermembrane space. And then there's a little bit of information about that. You might need to look that up for our conclusions. You're going to use a range of 2 to 9 with multiple readings from 3.5 to 8. Okay, this range should help you find the optimal pH of the intermembrane space to make as much ATP as we can. That's what we mean by optimal. We're going to record data on the worksheet, and then we're going to make a graph. So we go back to our worksheet. We continue in our worksheet by moving to the cellular respiration simulation. We complete this table that looks at changing the pH and focuses on how much oxygen is consumed, how much ATP is made. Those are important, both of those, okay? And then we also run this with and without something called DNP. So you have to learn about what DNP is. You're going to run this simulation with and without it. You can see that on our simulation site again. And then you need to draw a graph to show the effect of your variable on ATP production. Our variable is a change in the pH, all right? And so I didn't write that in there, but we're changing the pH of the intermembrane space. That's the independent variable. The dependent variable is ATP production. How much ATP are we making in moles? Okay, you're going to make a beautiful graph using Excel, just like you did in the first in the photosynthesis graph, and make a nice caption and answer some questions regarding the results of this cellular respiration experiment or simulation. And then there's a set of questions called applying your knowledge. Right, so we need to go back to our worksheet and check out what those are all about. Okay. So applying your knowledge There's a little bit of information here that you need to read about, a graph to look at that focuses on time over millions of years before present, and like the amount of oxygen, the amount of carbon dioxide in the atmosphere, okay? And then there's some information you're given. First about photosynthesis, and then second about respiration. And you have to come up with a hypothesis and an experiment. So the information we're given has to do with the temperature. The temperature apparently increased dramatically between the Triassic and the Jurassic periods. We see a big increase in temperature. We also see a change in which plant species dominated the world. Ferns become much more abundant when temperatures rise. The ginkgos decline. So in the Jurassic, there were hotter temperatures and more ferns than in the Triassic, okay? Now the question is, I wonder why, all right? I want you to think about the shapes of the leaves and what a fern leaf feels like. A ginkgo is a very thick, solid leaf. It's all one unit. There's no little gaps or air holes in it, like with a fern, right? And now you have to change the temperature under the photosynthesis simulation again and record the results of the temperature change. So you're going to go to your worksheet and your simulation. I'm going to scroll way down to where we are. Yes, here we are. The ginkgo and the fern photosynthesis experiment. You're going to look at photosynthesis under two different temperatures, thinking about how these temperatures affect photosynthesis. Then you're going to propose a hypothesis for why ferns increased when it got hotter, all right? So we have to think about what happened when it got hotter. Is it easier to produce glucose? Is it harder to produce glucose? That's what we're thinking about, okay? And then you have to propose an experiment to test your hypothesis. And I just want you to remember here, when you're making an experiment, you should not have a sample size of 1. You should not say, I'm going to plant 1 fern and 1 ginkgo. And remember, you need to put ferns and ginkgos in both temperatures and measure, say, growth rates or something like that. And then there's one more thing you need to do, and that's another applying your knowledge with respiration. So we have to go back again to our document, read about what happened during the transition from the Carboniferous to the Permian period. Oxygen levels go way up. So oxygen levels are really high during this period. Lots of oxygen. Also during this period, there were these enormous insects. Some of them were like, 12, having a wingspan of 28 inches, 2.5-foot insect. Okay, that's enormous. And the question is, does the amount of oxygen available have any relationship to the size of the insect? If you have more oxygen, are you going to be able to grow bigger? Maybe. So we want you to go to the cellular respiration simulation site and change the amount of oxygen. So change oxygen conditions and record your data in the worksheet. So you're going to record 100% oxygen and 50% oxygen conditions. I think because I know oxygen is required for cellular respiration, but the more oxygen you have, the more ATP you're going to make, right? And then think about this. Think about these insects that grew to be very big. How could they grow so big? Well, there was a lot of oxygen around. If there's a lot of oxygen around, maybe they can make more ATP, and that will encourage growth. That would be a hypothesis you could make. You need to make a hypothesis and make an experiment to test your hypothesis, but the experiment has to be one you can actually do. You can't say, well, I'm going to go back in time and work with these giant insects. Although that would be really cool, we can't do that. So you may have to think about raising insects in different conditions, insects in today's world, in different oxygen conditions or something like that. And again, sample size shouldn't be 1, all right? When you're all done, convert this into a PDF and turn it in. Remember, you'll have 2 Excel graphs in this worksheet. So this is a long worksheet. This is going to take a while. You'll want to get a good start on this early in the week and, work with friends. Talk to each other about your hypotheses and your experimental designs. Make good ones. They're worth a significant amount of points, okay? All right, I think that's everything, and for now, and I'm going to turn this video off because I'm tired of talking.